cellular aging in dogs operates independently of chronological age. Two dogs born the same year, living in the same environment, receiving comparable nutrition, can exhibit divergent rates of biological decline. One maintains mobility and cognitive function into advanced age. The other shows progressive deterioration in both domains years earlier. The variable isn't luck. It's not entirely genetics. It's the rate of cellular energy system degradation occurring at the mitochondrial level. And the process begins years before clinical symptoms emerge or standard diagnostics detect abnormalities. Mitochondrial function and energy production. Mitochondria convert nutrients and oxygen into ATP through oxidative phosphorylation. This biochemical process sustains muscle contraction, cardiac function, neuronal activity, immune response, tissue repair, digestive efficiency, and cellular maintenance. Each cell contains hundreds to thousands of mitochondria operating continuously from birth. Mitochondrial density varies by tissue type, with metabolic demand influencing cellular mitochondrial content. Performance of these systems declines over time in all mammalian species studied. In canine brain aging models using beagles, mitochondria isolated from older dogs demonstrate increased reactive oxygen species production and reduced respiratory capacity compared to mitochondria from young dogs. This is measured decline documented through direct biochemical assay of mitochondrial function, not theoretical risk extrapolated from other species. The rate of decline varies by tissue type, by breed, by individual metabolic history, and by environmental factors not yet fully characterized. Oxidative stress and cellular damage energy production generates reactive oxygen species as an inevitable metabolic byproduct. At low concentrations, ROS participates in normal cell signaling pathways and immune function. At elevated concentrations, it damages proteins, lipids, mitochondrial DNA, and nuclear DNA. Young organisms maintain equilibrium between ROS production and antioxidant defense systems through enzymatic mechanisms including superoxide dismutase, catalase, and glutathione peroxidase. This equilibrium deteriorates with age. ROS production increases per unit of ATP generated. Antioxidant enzyme activity decreases. DNA repair mechanisms lose efficiency, as documented in canine aging models. Damaged proteins accumulate. The accumulated oxidative damage in brain tissue manifests as cognitive decline, disorientation in familiar environments, altered sleep-wake cycles, house soiling despite prior training, reduced responsiveness to previously learned cues, and changes in social interaction patterns. These behavioral changes reflect underlying neuronal damage, synaptic dysfunction, and loss of cellular energy capacity in specific brain regions. In cardiac tissue, mitochondrial dysfunction correlates with reduced contractile efficiency, exercise intolerance, and increased susceptibility to arrhythmia. In skeletal muscle, it manifests as sarcopenia, reduced stamina, and prolonged recovery time following physical activity. These are not separate age-related diseases. These are tissue-specific manifestations of the same underlying cellular energy decline. The detection problem. Standard veterinary blood work measures established disease markers: hepatic enzymes indicating liver damage, creatinine and BUN, indicating kidney function, complete blood count detecting anemia or infection, thyroid hormones, glucose, electrolytes. These panels do not quantify mitochondrial efficiency. They do not detect subclinical oxidative damage. They do not measure cellular energy production capacity or ATP synthesis rates. Blood chemistry can remain within laboratory reference ranges, while cellular function has declined substantially from individual baseline. Reference ranges are population-based. They reflect the distribution of values in a sampled population, not optimal function for an individual animal at a specific life stage. A dog with declining mitochondrial function may show blood values in the normal range because compensatory mechanisms are still operational. Increased respiratory rate compensates for reduced oxygen utilization efficiency. Increased heart rate compensates for reduced cardiac output per beat. Behavioral adjustments compensate for reduced stamina. Symptoms appear when compensatory mechanisms exhaust their capacity. By that point, the underlying cellular deterioration has been progressing for months or years. Routine screening protocols do not measure mitochondrial endpoints, oxidative stress biomarkers, or subclinical bioenergetic decline in tissue samples. This represents a structural characteristic of current diagnostic frameworks in companion animal medicine, not merely a technical limitation. Evidence from long-term caloric restriction studies. A 14-year study of 48 Labrador retrievers compared a control-fed group with a group receiving 25% caloric restriction while maintaining complete and balanced nutrition. Pairs of litter mates were assigned to control or restricted groups. The restricted group received 75% of the food intake consumed by their control-fed siblings from 8 weeks of age through the end of life. The restricted group showed median lifespan extension of approximately 1.8 years compared to controls. Onset of age-related disease signs, including osteoarthritis, appeared later in the restricted group. Body composition differed significantly between groups. The restricted group maintained leaner body mass throughout life. Serum triglycerides, insulin sensitivity, and glucose metabolism differed between groups at multiple time points. This study demonstrates that metabolic parameters, body composition, disease onset timing, and lifespan outcomes correlate with sustained differences in caloric intake when applied consistently over the lifetime. The mechanism involves reduced metabolic stress, lower oxidative damage accumulation, improved insulin sensitivity, and reduced systemic inflammation associated with excess adiposity. Similar outcomes have been documented in controlled studies across mammalian species, though effect sizes and mechanisms vary by species and study design. Dietary intervention and mitochondrial markers. Research in aging dogs demonstrates that dietary supplementation with antioxidants and mitochondrial cofactors correlates with changes in specific mitochondrial parameters. In one canine model using aged beagles, an antioxidant-enriched diet containing vitamins E and C, mitochondrial cofactors, lipoic acid, and other compounds reduced AROS production and improved NADH-linked respiration measures in brain mitochondria compared to control diet. The dietary intervention group showed selective improvement in mitochondrial function parameters, while behavioral enrichment alone did not alter the same mitochondrial biomarkers. Behavioral enrichment, consisting of social interaction, novel toys, exercise, and cognitive tasks, improved performance on cognitive testing and reduced behavioral signs of aging, but did not change mitochondrial AROS production or respiratory function in isolated mitochondria. This demonstrates that different interventions affect different biological endpoints. Metabolic interventions influence cellular energy production pathways and oxidative stress markers. Environmental and behavioral interventions influence cognitive reserve, neuroplasticity, and behavioral outcomes. Neither category of intervention substitutes for the other. The biological systems involved operate through distinct mechanisms. Population-level data collection. The Dog Aging Project represents a longitudinal observational study enrolling tens of thousands of companion dogs to identify genetic, environmental, and lifestyle factors associated with variation in health span and lifespan. The study collects data on breed, body size, diet, exercise patterns, veterinary care, living environment, health events, and causes of death across a large population of privately owned dogs. The goal is population-level mapping of factors that correlate with healthy aging versus accelerated decline, and to support observational analyses and enable specific clinical trials, including interventions such as rapamycin, nested within the larger cohort. As of 2026, data collection continues. Initial findings have been published on relationships between body size and lifespan, genetic markers associated with age-related disease, and survey-based assessment of cognitive decline prevalence. Large-scale longitudinal studies provide observational data on real-world outcomes that controlled laboratory studies cannot replicate. Biological reality of cellular aging dogs age through progressive decline in cellular energy production, accumulation of oxidative damage to macromolecules, reduction in repair system efficiency, loss of mitochondrial quality control, accumulation of senescent cells, chronic low-grade inflammation, and gradual loss of homeostatic reserve capacity. These processes are measurable at the molecular level in tissue samples before they manifest as clinical disease detectable through physical examination or standard diagnostics. Chronological age measures time elapsed since birth. Biological age reflects the functional state of cellular systems, tissue integrity, and physiological reserve. Two dogs of identical chronological age can differ substantially in biological age based on cumulative metabolic history. This divergence is driven by the interplay between genetic predisposition, lifelong metabolic load, environmental exposures, and the efficiency of cellular maintenance pathways. The rate at which these processes unfold is not uniform across individuals, nor is it predictable solely by breed or size. Instead, it is shaped by the unique history of each animal, its diet, activity level, stress exposure, and overall metabolic health. Cellular decline precedes clinical detection by years. Chronological age measures time elapsed. Biological age reflects functional capacity of cellular systems. Standard diagnostics detect established pathology, not subclinical bioenergetic dysfunction. The gap between measurable cellular dysfunction and detectable clinical symptoms represents the period during which biological systems retain the greatest capacity for response to metabolic intervention. This interval narrows as cellular damage accumulates. Mitochondrial function continues to decline. Oxidative stress continues to damage cellular components. Repair mechanisms continue to lose efficiency. The cellular clock operates continuously. It does not pause. It does not reverse. It measures the progressive loss of cellular energy capacity and the accumulation of molecular damage that defines biological aging at the most fundamental level. The trajectory of this decline is influenced by factors such as caloric intake, body composition, oxidative stress load, and genetic predisposition. Some of these factors are fixed. Others are modifiable. The caloric restriction study in Labradors demonstrated that sustained metabolic intervention over a lifetime can extend both lifespan and health span. Most companion dogs do not receive interventions of this type. Most age at rates faster than their biological minimum. The window during which intervention can alter trajectory is finite. Cellular aging in your dog is occurring now. The rate is not fixed. The outcome is not predetermined.